Good evening and welcome everyone. Our presentation is about interferons presented by me, Aya Ammar, and my colleagues and supervised by Dr. Ahmed Abdullah. Our goal today is to give you some background about interferons. First, we will give you an overview about interferons. Secondly, we will talk about the pathway of induction. Then we will talk about the classification of interferons. After that, we will discuss the therapeutic application of interferons. Then we will talk about the uses of interferons in TB. And lastly, we will talk about the interferon alpha adjuvant activity. An overview about interferons. They are chemical signaling low molecular weight 20,000 to 30,000 daltons extracellular glycoproteins synthesized by different immune cells. They participate in the immune system, stimulating the responses against viral infection. Their name interferons is derived from their ability to interfere with the viral replication. They were first described members of cytokines. Interferon is secreted by a cell in response to the viral infection. Also, they have immunoregulatory function. Discovery of interferons. The role of interferon was first described in 1930s by Hoskin. A few years after Hoskin, many experiments find out about the role of interferons and try to describe it, but they were only confirmed in 1957 when Isaacs and Lindenmann, through cell culture research, demonstrated that cells infected by a virus had the ability to produce a protein that could make other cells resistant to other viruses. Interferons are classified into three types, which are type 1, type 2, and type 3. This video shows the pathway of induction. Once a virus infects a cell, it enters inside the cell, releasing its genetic material inside the cytoplasm. These foreign genetic material inside the cell start to synthesize viral product that stimulate the cell to make interferons, which are the red dots in this video. The interferons are secreted outside the cell and bind to cell surface interferon receptors of neighboring cells, which signal the synthesis of antiviral proteins. The antiviral proteins make the cell resistant to viral infection in several ways, including blocking the viral replication by breaking down the mRNA and stopping protein synthesis. Thus, a virus can enter the cell, but completion of the viral replication cycle is prevented. And now I will let my colleague to continue.
Good evening, everyone. I am Samra Suhail, third year medical student, and in this seminar we're going to discuss the three types of interferons. And I'm going to talk about type 1 interferons. Type 1 interferons divided into two types. As we are seeing here at the left side, we have interferon alpha and also we have interferon beta. Let's start with the structure. Several classes of interferons exist, and they have some similarities. For example, all type 1 interferons display the same characteristic structure of a helical cytokine, with a bundle of 4 up, down alpha helices, and in addition, alpha helix. In addition, almost all interferon classes are acid stable to pH2 and are heat stable. They are gene-like introns and they are closely conserved in length from 161 to 167 amino acid, with the protein sequence about 75 to 99% amino acid sequence similarities. Let's move on to the encoding gene for type 1 interferons. Gene encoding for type 1 interferons are clustered on human chromosomes 9p22. The gene responsible for the interferon alpha synthesis come in 13 subtypes like interferon A1, 2, 4, etc., while interferon beta is encoded by interferon beta 1 gene. In this slide, we have a figure that shows type 1 interferons receptor. The interferon alpha beta receptor is virtually a very common membrane receptor which binds endogenous type 1 interferon cytokines. This figure shows the major signaling pathway for type 1 interferons that we are going to discuss briefly. Toll-like receptors 7 and 9 are highly expressed on PDCs, and depending on the chemical properties and formulation of the ligand used and on the cell type activate signaling pathway involving either IRF7 or IRF5 and NF-κB. The IRF7 pathway preferentially induces a transcription of type 1 interferon gene family. Other receptors able to induce a production of type 1 interferon are toll-like receptor 3 and 4 signaling through the adapter molecule TRIF and the kinase TBK1. The IRF7 is acquired for optimal production of type 1 interferon induced by all these receptors, whereas IRF3 is essential only for the response to toll-like receptor 3 and 4. In this slide, we have 4 figures that show a specific action of type 1 interferons. Let's start with figure A. Figure A shows activate the expression of interferon-inducible gene that makes cell more sensitive to detection and elimination of incoming virus. Figure B activates cell intrinsic apoptosis and the expression of ligands that allow NK cell and CDOs to distinguish infected cells. C. Clonal expansion and differentiation of CD8 cells. Figure D potentiates dendritic cell immune response toward virally infected cell via toll-like receptor type 1 interferon signaling. Here we have some associated diseases with type 1 interferons like intestinal barrier disease, inflammatory bowel disease, colic disease, psoriasis, systemic lupus erythematosus, multiple sclerosis, and also we have cancer. And now I'm going to let my colleague Zakaria to continue.
Good evening, doctors and colleagues. I am Zakaria Mahmoud and I'm going to present type 2 interferons group. At the beginning, some basic information. Firstly, the sole representative of this group is interferon gamma, or as it's referred to as immune interferon. Secondly, there is no homology with type 1 interferons, so interferon gamma is unrelated to type 1 interferons at both genetic and protein levels. Thirdly, interferon gamma is produced by natural killer cells, antigen-presenting cells, cytotoxic T lymphocytes, and T helper 1 cells subset. That's to say, interferon gamma has a prominent role in both immune responses, the non-specific and the specific. Lastly, it is more immunomodulatory than antiviral. This means interferon gamma resembles interleukins in behavior more than type 1 interferons. Starting with the structure of interferon gamma. The biologically active interferon gamma molecule is a non-covalent homodimer formed by self-association of 2 major polypeptides in an antiparallel orientation. As it appears on the screen, each subunit consists of 6 alpha helices that comprises 62% of the structure, held together by short non-helical regions. The major molecule composed of 143 amino acids, and it's important to mention that little, if any, monomer is detectable in the serum, since only the dimer can display interferon gamma biological activity. Now we move to talk about interferon gamma receptor, a single type of interferon gamma receptor that is ubiquitously expressed on all cells except the erythrocytes. Interferon gamma receptor is composed of 2 structurally homologous polypeptides named interferon gamma receptor 1 chain, which is the internal brown chain on the screen, and interferon gamma receptor 2 chain, which is the external gray chain on the screen. Both interferon gamma receptor chains lack intrinsic kinase activity and must associate with signaling machinery for signal transduction. In terms of gene location on human chromosome, interferon gamma encoding gene is localized to chromosome 12 on the long arm region 15. while interferon gamma receptor chains encoding genes are found on separate chromosomes, where interferon gamma receptor 1 chain encoding gene is localized to chromosome 6 on the long arm, region 23, band 3, while interferon gamma receptor 2 chain encoding gene is localized to chromosome 21 on the long arm, region 22, band 11. On continue to the next subject. Now the scenario is the cells of source are stimulated or activated to produce interferon gamma. Then these molecules are going to bind to the receptor on cell surface. Now the next step is the signal transduction. Interferon gamma action is mainly through Janus kinase signal transducer and activator of transcription pathway, which is abbreviated as JAK-STAT pathway. This paradigm clearly shows interferon gamma, which appears in red, and interferon gamma receptor before receiving its molecule, of which the internal yellow chain is attached to JAK1, which appears as a blue elliptic, and the external green chain is attached to JAK2, which also appears as a blue elliptic. Now, whenever interferon binds and induces the dimerization of the two receptor chains, this leads to the activation of the Janus kinases, which resultantly activates STAT1 homodimer and interferon regulatory factor 1. Now, the phosphorylated STAT1 and interferon regulatory factor 1 translocate to the nucleus, where they bind to specific promoter regions called interferon gamma activation site to initiate or suppress transcription of interferon regulated genes. On moving to the next subject, I'm going to say that interferon gamma has an extending list of biological actions, of which the first one is macrophage activation. Interferon gamma is the primary cytokine involved in macrophage activation by the classical pathway. The activated macrophages highly produce pro-inflammatory cytokines, promote more intensive pinocytosis and phagocytosis, and improve microbial killing ability. The second action is the influence on antigen-presenting cells, of which are the dendritic cells. Interferon gamma directly stimulates antigen processing and presenting via increasing the expression of MHC class 1 and 2 molecules. Thirdly, the ability of interferon gamma to enhance the expression of cell surface adhesion molecules such as intercellular adhesion molecule 1, endothelial leukocyte adhesion molecule 1, as well as MHC class 1 molecules, consequently serve to expand and amplify the overall inflammatory response. Interferon gamma stimulates the differentiation of T helper 1 and cytotoxic T lymphocyte cells, while inhibits T helper 2, T helper 17, and T regulatory cells. Interferon gamma also influences B-cell mediated response as it regulates immunoglobulin isotype switching on B cells directly by antagonizing T helper 2 derived interleukins that induce MHC class 2 expression on B cells, and indirectly by regulating the development of specific subsets of CD4 plus T cells. Last but not least, T helper 1, T helper 2 cross-regulation. The opposing effects of interferon gamma and interleukin 10 serve to cross-regulate the development of specific immune responses. To furtherly explain this process of cross-regulation, it is stated that the release of interferon gamma is promoted by interleukin 12 and interleukin 18. Then interferon gamma inhibits the proliferation of T helper 2 cells and their cytokine production, including interleukin 10. As a result, a cell-mediated immune response is developed. While interleukin 4 and interleukin 10 are the negative regulators of interferon gamma production, interleukin 10 inhibits interferon gamma synthesis by T helper 1 and natural killer cells, thereby inhibiting the development of cell-mediated immunity, and at the same time interleukin 10 actions promote the development of a humoral immunity. Lastly, we are going to discuss the role of interferon gamma in autoimmune diseases. Now here it comes a split. One side titles that interferon gamma is implicated in the problem, and they stand on the fact that interferon gamma activates immunoglobulin G Fc receptor and complement, which contribute to disease severity. That interferon gamma increases anti-double-stranded DNA and antihistone autoantibodies, and that interferon gamma promotes B-cell immunoglobulin G class switching to more pathogenic autoantibodies such as immunoglobulin G2A and immunoglobulin G3 in mice. On the other side of the disputation, some studies claim that T helper 17-driven experimental autoimmune encephalomyelitis EAE, experimental autoimmune uveitis EAU, and collagen-induced arthritis CIA may be prevented by interferon gamma. Since interferon gamma inhibits T helper 17 cells differentiation, depletion of endogenous interferon gamma leads to increased number of interleukin 17 producing cells and aggravates the disease, especially in EAE and CIA. So these studies address the protective effect of interferon gamma, while other studies contradict. Thank you for listening, and I'm going to hand the mic to my colleague Hazem.
Good evening. My name is Hazem Shamil. I'm a third-year medical student, and I'm going to talk to you about type 3 interferons. I'm going to start with the definition and some general information about type 3 interferons. Type 3 interferons, also known as interferon lambda, are the latest addition to the interferon family, discovered in 2003. They have functions that are similar to type 1 interferons. The type 3 interferon family consists of 4 cytokines or 4 representatives. The first one is interferon lambda 1, also known as interleukin 29. The second and third ones are interferon lambda 2 and interferon lambda 3, known as interleukin 28A and interleukin 28B respectively. And the last one is interferon lambda 4. They are produced primarily by cells of epithelial lineage and neutrophils, and they are also structurally related to cytokines of the interleukin 10 family. Type 3 interferon receptor, also known as IFN lambda R. It's a heterodimeric receptor composed of 2 different chains. The first chain is the interferon lambda R1 high affinity chain, shown on the left side of the diagram, and the second chain is the interleukin 10 R beta low affinity chain, present on the right side of the diagram, and this chain is shared with other cytokines. This receptor is mostly expressed in epithelial cells and neutrophils, and is shared between all the members of the type 3 interferons family. Type 3 interferons gene locus. The interferon lambda encoding genes for the 4 members of the interferon lambda family all exist in the long arm of chromosome 19. The interferon lambda 1 gene is located downstream of the interferon lambda 2 gene, and the interferon lambda 3 gene is located downstream of the interferon lambda 4 gene, as shown in the diagram. Now I'm going to talk about the biological activity of type 3 interferons. Type 3 interferons work inside the body mainly in 3 ways. Firstly, they prevent viral replication in epithelial cells. Secondly, they modulate the immune response in its infancy, and work as pro-barrier cytokines directly by affecting neutrophils to downregulate the production of granules and reactive oxygen species that damages the tissue and compromises the epithelial barrier for further viral infection. This can be summarized briefly as limiting inflammation. And thirdly and finally, they stimulate the adaptive immunity indirectly by influencing the dendritic cell's function to promote T-cell memory, therefore favoring long-term protection and less damaging immune response. Now I let my colleague to continue.
Good evening. I am Aya Almar. I will talk about therapeutic applications of interferons. Interferons and interferon inducer are available commercially. Interferon alpha is rapidly absorbed after both intramuscular and subcutaneous injection. This rapid absorption combined with a short half-life means that frequent injections are needed to maintain adequate concentrations in the body. Polyethylene glycol PEG to enhance their half-life and make once-weekly dosing possible, and reducing side effects by having a predictably lower peak concentration of the exogenous cytokine. In this slide, I will talk about use of interferons in human papillomavirus HPV. Topical interferon inducers like Zaclara and imicomod. The 5% imicomod cream should be applied to affected areas 3 times a week for up to 16 weeks, while the newer 3.75% cream Zaclara can be applied once daily for as little as 8 weeks to treat external genital warts caused by HPV. Recurrent respiratory HPV infection has also been effectively treated with interferon alpha. Imicomod 5% cream has been used successfully in the treatment of the plantar wart, a smoother, flatter manifestation of HPV 1, 2, and 4 on the foot. Systemic PEG interferon alpha and a topical retinoid has been reported for mucosal carcinomas due to persistent and widespread HPV infection of the skin. Now my colleague Alpha will continue.
Good evening. Welcome doctors and colleagues. I'm Ofa Haitham and I'm going to continue to talk about therapeutic applications. Interferon alpha-2A, Roferon A, is used in the treatment of hairy cell leukemia, AIDS-related Kaposi sarcoma, and chronic myelogenous leukemia. Interferon alpha-2B is used in the treatment of hairy cell leukemia, malignant melanoma, condylomata acuminate, AIDS-related Kaposi sarcoma, chronic hepatitis C, and chronic hepatitis B. Interferon alpha-N3, Alpheron N, is used in the treatment of genital and perianal warts caused by human papillomavirus. Interferon gamma-1b, ACT-immune, is used in the treatment of chronic granulomatous disease and severe malignant osteopetrosis. Interferon beta-1b, beta-seron, and interferon beta-1a, Avonix, is used in the treatment of multiple sclerosis. Lastly, pig interferon beta-1a or Plegridy is used in the treatment of multiple sclerosis. Now I'm going to talk about interferons and COVID-19. The new coronavirus, SARS-CoV-2, inhibits interferon production in the upper airways. This weakens the immune response and helps the virus to survive. But when the virus reaches the lower airways, there is an exuberant immune response, including an increase in type 3 interferons that may be harmful. Where evidence from Italy suggested that type 3 interferons increase the risk of life-threatening bacterial superinfections in the lung, even though recent evidence suggests that type 3 interferon lambda, can fight viral infection and limit inflammatory damage. Here my colleague Hussein will continue the subject.
Good evening, I am Hussein Talal, third year medical student, and I am going to continue the therapeutic application of interferons and talk about interferon in chronic infection with hepatitis B and C. Regarding to hepatitis B, we use interferon to decrease viral replication, leading to the prevention of a progression to cirrhosis or hepatocellular carcinoma. And regarding to hepatitis C, interferon stimulated genes are induced by interferon and disrupt viral replication. Ribavirin is used in combination with PEG interferon administered as a subcutaneous injection once weekly for duration up to 48 weeks as the standard for treatment of HCV. Use of interferons in tuberculosis. Interferon gamma is used as an alternative treatment of drug-resistant TB, as aerosol administration safely delivered to the lower respiratory tract without systematic side effect. Current studies are being done on the uses of interferon gamma in intramuscular or subcutaneous administration in MTB-resistant patients with mostly positive results. Interferon gamma release assays IGRAs are used as faster way of identification and diagnosis of latent TB. Interferon alpha adjuvant activity potentiate response to intramuscular influenza vaccine. Oromucosal administration of recombinant interferon alpha provide immunity against viral infection and tumor cell growth. None or low responders to a previous vaccine showed an improved immunoglobulin response with a recombinant interferon alpha and HPV vaccine. Other classes of interferons as adjuvants show less promising result as that of interferon alpha. Summary. Interferons are species-specific proteins that induce antiviral and antiproliferation responses in a human cell. There are three types of interferons: type 1, type 2, and type 3. Interferons are produced from different cells in the body. Interferons participate in pathogenesis of many diseases. Interferons uh, have many therapeutic uses, including HPV, HCV, COVID-19, TB, and etc. For more details, these are our references, and you can ask us if you have any question. Thank you for listening.